Back. Okay, well, Evan, first of all, thank you very much for agreeing to do this session. Maybe you want to quickly introduce yourself before starting, and
Sure.
then that'll be great.
Hello, everyone. I'm sorry I'm not there. My name is Evan. I am a researcher here in the Faculty of Medicine. My specialty is in molecular neuroscience. I study in particular neurodevelopment and autism. And I've been here in the faculty since 2011, since it started 10 years ago. So I just want to say, I see, Michael, that there's two people in the room with you right now.
There's two people in the room. There's one person online, and there's two more people that should arrive in a few minutes. So it's
okay.
And committee.
And everyone
we're
here.
Expecting. We're a group of 7.
And everyone.
Sorry, you're cutting off again.
And everyone here is medical doctors, if I understand.
Yes.
Okay, very cool. Okay, so I also teach a course in scientific writing in Faculty of Medicine. So what I tried to do now is to make sort of a shortened version, an overview of what I teach in regarding to writing a manuscript. Again, so go through different parts of a manuscript. Now, this is very much an overview, and if there's some parts I don't know, maybe we've talked about earlier in the course, you can let me know, hey, we should skip over this, or if there's some things that maybe I'm not talking about, you think, hey, you really have some important questions, just butt in, let me know, whatever. So what I thought to do is I just wrote down here, as you can see, to start with some writing tips, things that I see as just go, whenever I edit students' papers or any of these papers, these are things that repeat themselves all the time, very important things about writing, and just basically go through manuscripts, what a manuscript should look like, what are its different sections, what should be in each section, how to best write the manuscript so that the reader can understand it, because that's the most important thing you want, is that the reader will come away really understanding what you write, also that the reviewers will understand what you write, because that's also important to talk about. So we'll go through some writing tips, shortly talk about introduction. I know you've talked about introduction in other contexts, so very shortly. Then about what goes in the results section, discussion section, materials and methods section, abstract. And at the end, talk a little bit about the process of getting a manuscript ready, of submitting manuscripts to a journal. And I just thought this morning, I don't know if you talked anything about how you put references in a paper, maybe not or yes. If not, then I can also talk a few minutes about that as well. So again, I have here sort of an overline, and if there's certain things that I also see if I need to skip over some things, because what we talk about in the end is particularly important in terms of journal submission. I brought here something which comes from a very important book. You see here called On Writing Well. It's a book from 1976, very, very famous book about how to write well, obviously. This is written about 50 years ago, but not much has changed since then. And I wrote this because this is a very important paragraph where she says, or he says, the secret of good writing is to strip every sentence to its cleanest components. In other words, rather than read the following, every word that serves no function, every long word that could be a short word, every adverb that carries the same meaning that's already in the verb. For example, yellow sun. Every passive construction that leaves the reader unsure of who is doing what. These are the 1,001 alterants that weaken the strength of a sentence, and they usually occur in proportion to the education and rank. So in other words, the more that a person becomes educated, the more he wants to write the really elaborate, long sentences, and that's really, really bad because it makes the readers not understand what you're writing. So the number one thing they try to teach the students is to make your writing as short, concise as possible. And that's the way they'll be understood by the reader. So for example, just to give an example, when you teach a course, so at the end of every lecture, I give the students something to edit and to make shorter, something that could have been published or someone else's writing. So just like to give an example, sort of a stupid example, but it's an example. Look at the following sentence, right? Everyone in the room there can read what's on the screen, right?
Yes.
Yes, okay. So I would like to claim that the author should be considered to be stupid. Okay, that's a sentence. How can I shorten to the?
I think the author is stupid.
I think the author is stupid. Okay, I'm gonna write that again. I think Wait, just 1 second. So, is someone speaking right now? No. Okay, I'm hearing strange voices. No, just a second. Okay. I think the author is stupid.
You're hearing the voice of the psychiatrist walk.
What was that?
You were hearing voices, and then the psychiatrist just walked in.
Okay, I think the author is stupid. So that's one possibility. So one possibility is the author is stupid. Right? Another possibility is, which is very similar to I think Michael just said, I claim that the author is stupid, right? Now, the thing is, it's very possible to just write the first thing, the author is stupid, and that's taking away all the unnecessary words. However, sometimes you don't want to say things which are too strong, which we'll talk about later. Therefore, the words I claim are often necessary when making a statement which is not proven fact, unless it's proven that the author is stupid. So again, it's just an example of shortening sentences, trying to look at every sentence, what word can be taken out. One of the reasons people do this, I've noticed, they write long sentences is because they are shy in their writing. In other words, they don't want to make any sort of strong claims, but don't worry about it. Don't be too shy in how you write, just say short, concise sentences, it's okay. Here I just give a few examples that you'll get these slides, take advantage of looking at different words that people use, shorten the meaning, number of, the same as many, just using shorter phrases, shorter words, make everything much more understandable for the reader. Because at the end of the day, imagine that your reader is going to receive some 4, 5, 6, 7-page manuscript and is going to have to try to read this, understand it. You want to make everything as simple and short as possible. Some more examples, I won't go through all of them, but as you can say, for example, in all cases, to say instead, always. There's actually one example here that I noticed that I make this mistake. I often say in order to, but you can just say to. So for example, in order to determine, you can just say to determine. Same thing, less words, more simple. One other thing I just mentioned from what she wrote as well, or what William wrote, I think it's a he. Every adverb that carries the same, oh no, sorry, not that one. Every passive construction that leaves the reader unsure of who is doing what, right? So I don't know if you talked about this previously, but there's generally a good idea, try to use active voice as much as possible in most of the manuscript. As we'll discuss later, there's certain parts of a manuscript that you must use passive voice, particularly materials and methods. But in other areas, it is often better to use an active voice instead of passive while writing. So just give an example here. You can say cigarette ads were designed to appeal especially to children. It's a fine sentence, that's good, that's right. The active voice would be, we designed the cigarette ads to appeal especially to children. What's important to realize is our linguistic brain is designed to understand active voice much easier than passive voice. We understand active voice much better, which means we should use it more. So this second way of saying it is better in terms of helping the reader understand it, but more than that, well, as you see here, the responsible party exists always in active voice. We designed the cigarette ads. And passive voice, you don't have the active party. Who did it? I don't know. The only way for me to know would be to add it at the end of the sentence. For example, by us, right? If I didn't add that, I wouldn't know. So therefore, in this particular case, or most cases, it is best to use active voice when possible. As I say here in the bottom, there are some cases where we don't want to identify responsible party, and therefore we use the passive tense. So for example, sometimes in a manuscript, for example, I might write cells were incubated with cytokines. Okay, that's fine. I don't want to say who did it. I incubated the cells by cytokines. I don't want to talk about myself. You usually don't refer to yourself in a manuscript. So therefore, some cases where passive voice is.
Evan, we have a question. One thing.
Yeah, sure.
Actually, I also thought that when you use the passive voice, you emphasize the action instead of emphasize that you did it. And another thing, I think everything that's done in the study is done by the authors, by the researchers. So why I need to emphasize every time that I did it?
A fabulous question. Thank you. So yes, you're right. And that's exactly what I was trying to get into the end, is that sometimes you do need a passive voice. Instead, you do not want to include yourself when describing results. You, and that's an important point, you are not the subject of the paper, right? So you do not want to refer to yourself. Sometimes maybe in a discussion it will be appropriate, as we'll talk about, but you're right, in particular results, you don't want to take yourself out, therefore use passive tense when describing what was done. However, if you can, and it's just what I'm saying here is that if you can use active tense, I would try to give some examples. Let's say I said cells were incubated with cytokines, okay, like I said before. So I described and I used passive tense. And afterwards I said, these cytokines induced cell proliferation. That's active tense. So I talk about the relationship between two things in a study, not myself, but two things in a study, then I use active tense. However, therefore, there I don't want to say cell proliferation was induced by the cytokines, right? Because that's passive. Now, that's also true, but I'm just saying that the active tense will be much better understood by the reader. So when possible, use the active tense, and it's usually possible when describing the relationship between two things within the study and not yourself. Does that make sense?
Yeah. Thanks.
No problem. And I think this actually points out well, and this next example, and this is the sort of thing that would be in an introduction of a manuscript. And again, I think comes on the same idea. So give an example of general dysfunction of the immune system has been suggested at the leukocyte level in both animal and human studies. So it'd be better to say both human and animal studies suggest that diabetics have general immune dysfunction at the leukocyte level. So again, I'm taking at the end, the animal and human studies, and I'm just putting it at the beginning and making it active tense. So as much as possible, use active tense. I use another type of example, which you'll find a lot in introduction of papers, is you might find something, let's say someone had the same sort of thing, but they didn't say in human animal studies, and they put a reference. The reference is, I don't know, Edelstein et al., right? That's inside a reference at the end of the sentence. So you can rewrite that sentence that says Edelstein et al., put that at the beginning of the sentence, and say, determined that. So when talking about someone else, not myself, someone else, I can put them at the beginning of a sentence as the active subject. You say, Edelstein et al. Determined that diabetics and general immune dysfunction at the leukocyte level. So I have to change the sentence a little bit to make it understandable. But the idea here is to, as much as possible, to try to use active tense, if that makes sense. One other, so what we said before, first of all, is trying to make everything as concise as possible in terms of writing tips here. Trying to use active tense as much as possible, except for when talking about something you did or materials and methods. Let's talk about later, which is always passive. And the last thing is in terms of constructing paragraphs. In terms of constructing paragraphs, I'll actually go to the next thing. The idea is that one paragraph is one idea. Okay, so every paragraph, before writing a paragraph, think of what is the idea that I want to have in that paragraph. And that is, and everything else in it should just be an explanation or developing that idea. And very often the first and last sentences of the paragraph might be reiterations of that idea. So for example, I showed you this paragraph before, right? And the very first sentence says, the secret of good writing is to strip every sentence to its cleanest components. That is the idea of this paragraph. Everything afterwards is explanation or is developing that idea. So when I always write, at least I always think of every paragraph as one idea that I'm going to develop. And then as I go to the next paragraph, the question is, what is my logical shift from the previous idea to the current idea? And I can develop further the overall idea of the manuscript. But again, I'm saying this now, but I'm going to show you in some introductions and results from a few other papers how this happens. So we'll go to that in a few minutes. So we're talking here about manuscripts, about published manuscripts after you've done your study. So the first point to get across to the scientific manuscript is a story. In other words, as I write here, many people think that telling a story is somehow unscientific and that you're supposed to show data and facts only. On the contrary, the manuscript needs to make the idea dateable, accessible to the reader. You're writing a story, therefore you want to think of it in terms of how to make all the data as accessible as possible. So very often, the way this is shown is as an hourglass figure, that if you have an introduction to a paper, an introduction is starting from a very, very general idea and is developing from that general idea to your particular scientific question. So for example, a general idea is, we do not know how to treat ovarian cancer. Your particular research question might be, does phosphorylation of the protein BDNF help develop cancer, the ovarian cancer, and can we treat that phosphorylation? So you have to get with an introduction from that very general idea and give all the information necessary to understand that very specific question. So you go from the general to the particular within the procedure or results as well. You're only talking about that very particular subject. And in a discussion, you're taking your finding and now bringing it back to the general question. So now that you've understood how phosphorylation of a protein might develop, help develop particular cancer, then you have to bring that finding back to, okay, what can this mean for treatment? How does this align with other studies, et cetera? So this is just an overall outline of how to look at the general story that you're trying to build. And then we're going to each part. So start with the introduction. I know you've talked about introduction before, so I'll just say a few things about it. Number one, and say here, the most important function of the introduction is to give the reader the information necessary in order to understand your research and to know what is the missing information in the field that you are providing. Anything which doesn't fall into one of these two categories should not be in the introduction. When you write a manuscript, you usually have a word limit for introductions. We'll talk about this later, but it could be 600 words, could be 500 words, could be 1,000 words. Depends on the journal you're submitting to. So you don't give some sort of overall introduction into the whole field. It's just a mistake, and readers are honestly not interested. So what you want, if they were interested, they would read a review. So what you want to give them is all the information necessary to understand why you did the experiment that you are presenting that you did. That's the overarching goal of the introduction. So a way to look at this is that you first of all start with your overall background, background in the general research field. What is the known information? What do we know today? And then you want to present at some point, however, It is not known that. What is the knowledge gap in the field? What do we not know? And of course, you're going to come and fill in that gap. So that knowledge gap should be in line with whatever your particular research question is. Followed by some sort of perhaps hypothesis, question. In other words, what will be in this paper that will help to answer that over question or fill in some of the knowledge gap. Now, when you look at different manuscripts, which we'll do in a minute, there are some that actually give in a lot of information about what they did in their paper. It's not necessary. You can do that, you can't do that, or you can decide not to. Everyone has their certain style. But what is important is that you have the full information, starting from the very general to the particular, and that you have what is the gap in knowledge, and ending with some sort of statement, some sort of understanding at least of how you're going to tackle this particular knowledge gap in the manuscript. So I'm actually going to give one example. To do that, I'll have to screen for a minute and go to, where is my screen? Here's my screen, share. So you can see a paper right now, right?
Yes.
Yes, okay. So here is a paper promoting social behavior with oxytocin and high functioning autism spectrum disorders, okay? So here they took people who have autism and they gave to them oxytocin to see how it affects their behavior, okay? So they start, of course, their paper with an abstract that you see in bold, and they have an introduction. Now, if you notice, introduction starts here and it ends here. I don't know how many words that is, but it is 1, 2, 3, 4 paragraphs. Most introduction manuscripts are around 4 paragraphs. There's not a rule, but that's the general thing. Again, I have to say one thing. As you can see, I come much more from the biological field. There are some fields that might be different slightly in their overall word counts, etc. So you have to keep that in mind. But 4 paragraphs is a good overall statement.
I cannot see the methods here, Ivan.
What was that?
I cannot see the methods between the production and the results.
Excellent. Every journal, we'll talk about this later, every journal has a different order for how they present the different sections of a paper.
Okay.
So PNAS, for example, this particular journal puts it at the end. And more than that, as you'll see here, let's look at results. I'm going through the results. Here they have results. Here they have discussion. Here, and after discussion, they have materials and methods. And materials and methods is actually, no, that's enough. So yeah, so here it's near the end. So there is no order per se. And also in terms of when you write a manuscript, right, it's not important what you write first, what you write last. I generally think it's the best to write results first, because that's what you know the best, and then start writing the introduction and discussion. The abstract is almost always the last because that's just summarizing everything. So if I go back to introduction, the first paragraph here is a very overall statement. We're not going to read everything here. Just you see is overall about our brain has a social ability, and in autism, they have problems in social behavior. And that's the whole idea of the first paragraph. So again, a very general knowledge about what is autism, what is social behavior. Whoops, sorry, that went away. And then you go, what's going on here? Maybe I have to, okay. So this second paragraph, very short paragraph, says it's unclear what causes autism. At the very end, says there's a hypothesis, maybe it has to do with oxytocin. Okay, so third paragraph is an introduction to oxytocin. Again, a very general statements. So, and the first paragraph is generally what is social behavior in autism. The third paragraph here is what is oxytocin. And then this fourth paragraph, which is longer, starts off with describing a previous experiment about what is the connection between oxytocin and autism. So now that we have introduced both separately, now he wants to bring them together and says experimental manipulation of brain oxytocin levels in healthy human subjects confirms its involvement in the expression of human affiliated social behavior. So here is now all of this is describing previous knowledge. So again, after we had the general description of his autism and oxytocin, now we get previous knowledge about what is the connection between them. But now we have the important part, which is now what is the missing knowledge. In the light of the above findings, a key question regarding both the role of oxytocin in the nervous system and the pathophysiology of social disorders and autism is whether administration of oxytocin, can influence social interaction behavior in individuals with autism. So now I have a sentence, sort of a long one, but this is the missing knowledge. And the rest of this paragraph is a description of how they will try to solve or to gain more knowledge about this unknown knowledge, about this question. So that is, I think, a good example of an overall outline of an introduction, where you start off with your general definitions of what is autism, what is oxytocin, etc. Then you introduce previous knowledge about the connection between them. Then you introduce what is unknown, what is the next step that is unknown, and finally, at least a few sentences about how this manuscript can possibly tell us important new information to close the knowledge gap. One other important thing I'll just mention here, that maybe noticed already, but in any case, notice that in here, it it makes a statement, it references a previous paper, and then puts here the reference. That's reference number 17. You always reference a manuscript in the first sentence that you mention it. Even if you talk the manuscript, even if you talk about the manuscript for 20 sentences, you reference it at the end of the first sentence. This is very important. I see a lot of people make that mistake where they wait till the end to put a reference. You always put a reference at the end of the first sentence. And then you can continue more to talk about that manuscript if you want without referencing it again. That was my example of introduction. Are there any questions about introductions in general? No. So I'm going to go back for 1 second. I'm going to go to results. Just a second. Go back to the Zoom. Your screen. Maybe I'll stop share. Very good. Okay, share screen. Then share. Okay, so now you see again the PowerPoint, right?
Yes.
Cool. Okay. So now we're going to go back to talking about the results. So now you have finished your introduction. You've introduced your subject. You're going to talk about what you found. So in the results section, what's important is we should summarize what the data show, describe the data, and point out important relationships in the data. And also talk about, and maybe at the end of a paragraph, a conclusion, or at least a sub-conclusion about what you have found. And you cite, of course, the figures and tables in the results. And as I write at the end here, avoid simply repeating the numbers that are already available in tables and figures. So I'm going to give you some examples here. Let's start with this. We have here a table. You have done a research on the difference between bad witches and good witches. Okay. And you see here, you have all sorts of characteristics. You have your N number, you have your age of your witches. You have how many were female, what is their BMI, systolic blood pressure, exercise, employment, are they smokers or not, right? So this is your information that you have gathered in your research, and now you want to write a results section about it. And this is a possible results section, okay? So you write the characteristics of the bad witches and good witches are shown in table 1. There was a significant difference in ages between the groups. The mean age of the bad witches was 45, and the mean age of the good witches was 36. There was no significant difference in gender between the groups, with the bad witches having 85% females and the good witches having 83% females. BMI was not significantly different between the groups, which both had normal BMIs. Systolic blood pressure and exercise were significantly different. The bad witches had a mean blood pressure of 140, whereas the good witches had a mean blood pressure of 120. Estimated daily exercise was higher in the good witches than the bad witches. Employment was not significantly different between the two groups. So first of all, what should I change? You can tell me what should I change in this results section?
Nothing. All of this can be replaced by putting the piece.
I didn't quite understand. I might have problems with connection. What was that again?
I said that all this paragraph can be like we can do like to be replaced by putting the p values.
Right. Okay. So you're saying something which is missing here. You're saying what is missing here is p values.
Yeah. And
okay. First of all, definitely agree. As we talk about later, there are some journals which demand that you write p-values in the description of the results section. Not all journals do that. Some do.
Okay.
But yeah.
So I mean, just to put it in the table, and all this paragraph we can like do without it, like.
So actually, you might not see very well from where you're sitting, but there is here, it says P is less than 0.05, and there's like asterisks where there's p-value.
Okay.
Okay, that's definitely true. So first of all, the first thing is that the first sentence, you do not need this first sentence at all. What do you do instead of that? You never say, our results are shown in figure 1, in figure 3. You don't write that. Rather, after the end of the first sentence, in parentheses, you cite the figure. So this is table 1, right? In parentheses, you put at the end. So that first sentence shouldn't be there. Number 1. Number 2, more than that, most of these numbers here should not be there. If they're in the table, and if I can see the numbers, I do not need I do not need to repeat numbers which I already see. Therefore, I gave an example here. You can write, this is a revised version of these results. You can see the witches were on average lean and predominantly female, table 1. The bad witches are significantly older, had higher blood pressures, exercised less, and were more likely to smoke than good witches. More bad witches were unemployed, but this difference did not reach statistical significance. So here I am not describing all of the numbers. Rather, I'm just describing what are the relationships that I see within my data. What are the important relationships that I need to, that are important to understand, right? I'm not describing all of the numbers. There may be some cases where you do want to write the number if it is really interesting. Let's say, for example, the systolic blood pressure of witches was 250. I don't know. So yes, you might want to write that because it's so amazing. Then you can write that in the results as well. But if not, if it's just some sort of difference and the number isn't really important, then you don't write the number. what I mentioned before is that there are some journals which would want you to write p-values in the results. So they might say bad witches are specifically older. You put in parentheses p equals 0.02, for example. That can happen, but that's dependent on journals, and you have to read the instructions of the particular journal before giving to them. So this is another example with basically the same idea, talking about running during pregnancy and how many women run during different trimesters of pregnancy. Won't go through it, but you get the general idea, where in the original results, they had a lot of numbers, what percentage of people were running at what time, etc. But when you edit it, you take out the numbers. I just want to understand the relationships. I want to understand what is the important relationships I should understand between these different numbers. Of course, this is assuming that these numbers are described in your table. So the reader can always look at the table if they want to understand the numbers. So in terms of organization of results section, if it's a very long manuscript, you should consider doing subsections, which is fine. You do not have subsections in introduction, but you can in results. And an important idea is to maintain a logical flow from one paragraph to the next. Why? And for the reader to always understand, why did you do a particular experiment? Then you describe that experiment. Then what I understand from that experiment. So actually what I want to do to understand that is actually go directly to a results section, which I think is a very good example. Again, I'm going to shop share, go back to share screen, share. Let's go to introduction, lecture number 3, this one. So this is a paper in a journal called Cell, which is a very high quality journal. And this is a paper where they looked at the fact that different gut microbiota can affect the synthesis and the metabolism of serotonin in the gut. Okay.
Can you put it in full size that we can really see?
Yes. Excellent. Thank you. Let me see. How do I?
It's in view, I think. You can put there full size, full screen.
But let's view, page display, single page view. Actually, let me try something else. I think if I can get rid of this, that helps. May somehow get rid of this, collapse all. No, that does not. Maybe I can do this.
The arrow, wait.
Like that? Woohoo. Thank you very much. Okay, now I actually have to go back a little bit. Where's my zoom in this thing? Here we go. Right. Okay, I think this would be the best. So again, this paper starts with a summary, in other words, an abstract. Then it has an introduction, which didn't go through this introduction, but then we go through results. So results starts here. I actually want to go through, there's some places I think I'm more, no, I didn't. There should be some, okay, I'll start here. Here, look how they start a paragraph. To identify the specific steps of 5-HT, this is serotonin metabolism, that are affected by the microbiota, key intermediates of the serotonin pathway were assessed in colons from germ-free and SPF mice. They tell me in the beginning of a paragraph, in order to determine something, this is what we did. And then we won't read through the paragraph, but the whole paragraph is describing what they found in terms of different intermediates, the serotonin pathway in the colons. Long paragraph. And then the last sentence is, after they describe all of the results, sorry. Where did that go? Okay. Okay. Collectively, these data support the notion that the microbiota promotes serotonin biosynthesis by elevating TPH1 expression in colonic ECs. So they begin the paragraph saying, in order to determine serotonin pathway changes in the colon, we looked at these intermediates. And at the very end, they tell me what they found. These data support the notion that the microbiota promotes serotonin biosynthesis by elevating TPH1 expression, TPH1 being a particular enzyme. And everything in the middle is just this description, very concise description of each of the levels of these results. Now, the next paragraph, they go down, begins the same way. To confirm that deficient serotonin levels in germ-free mice are microbiota dependent and further determine whether effects are age dependent, germ-free mice are conventionalized with an SFB microbiota at birth. It's not important for us right now to understand all the biology here, but the idea is, again, you start the paragraph with, in order to determine something, this is what we did. Then they describe the results. Right? And then at the end, see if I can move it over here. So they say overall, we demonstrate that microbiota mediated elevation of host serotonin is postnatal inducible. I can't read what's said here because of Zoom, and not dependent on the timing of host development. So we found that it is not connected to development, but is connected to microbiota. So again, an explanation, one sentence explanation of what my results tell me. So we start off with what do you want to know, then we describe a result, and at the end of the paragraph, what is the conclusion of my results? And if we were to go through this whole manuscript, that same paragraph, just a second, if we were to go through this whole manuscript, that same paragraph, that same building of a paragraph is found throughout the whole manuscript. To assess the reversibility of microbial effects in host serotonin metabolism, we depleted. That's what they did. They show the results. At the end, what are the conclusions? So they are going methodologically through the manuscript, through the results, from one idea to the next, starting every paragraph with what we did, and showing results. Then at the end, what can we understand from that? So we do throughout the manuscript, throughout the results, want some sort of mid-conclusion of what these results tell us. Again, not in a full paragraph, but every once in a while, one sentence, usually at the end of a paragraph. Does that make sense? Any questions about that? Makes sense. And if we go back, second, show something. Should be much easier if it wasn't by Zoom, but that's what we have. Let me see. So you'll notice that they have to mention what they did, but they don't describe in detail how you do it. That you leave for the materials and methods. That's an important difference. Materials and methods, you describe in detail the methodology, what you did, but in the results, you just explain enough for the reader to understand what you did without understanding the methodology. So I'll try to give one more example. And if we go back to here. So I'll give an example just from a sort of a fake experiment. Let's say I have an experiment. We want to understand why people in Wisconsin are overweight. I don't know if they are, but let's pretend. So you want to understand, is it because they go to McDonald's all the time? So here I have a results section where I start to understand if the increase in obesity seen in Wisconsin is due to increased visits to McDonald's. We determined how many times each individual in our Wisconsin study visited McDonald's for each of the last 10 years. Okay. And then I give a one-sentence explanation of how you did that. Because how do you know how many times you visited Wisconsin? Maybe you gave them a questionnaire. Maybe it's something else, but no. This was done by installing a GPS tracking system into the brain of each individual, followed by online tracking using Google Maps. So we know each person, if they went to McDonald's, by a chip found in their brain. Okay, so explain that. Because without that knowledge, I don't understand the results. So I have to explain what I did enough so I understand the results, even though I don't go through the technical methodology. If I'd done a questionnaire, I could just send here, this was done by a questionnaire. And then that's enough. And then the details about the questionnaire are found in the materials and methods section. A few things just keep in mind. Again, this may be repeated from things you learned about before, I don't know. But a few things to keep in mind about the results section. Do not forget to talk about important negative and controlled results. These are very important. So if you did a controlled experiment, for example, you also put a chip into the brain of people in New York to check as a control group if they go to McDonald's more. It's very important to talk about all controlled experiments in the results and explain them as well. And also about negative results. This is actually a general thing, which is very important, and explain what I mean. Again, I assume you know this already, but in any case, I sometimes have a student come and tell me, Evan, the experiment didn't work yesterday. So I ask them, okay, explain to me why it didn't work. And they tell me, oh, the results were negative. There was no change after the, after the treatment. So, okay, that's the experiment worked just fine. It just didn't give the results you wanted. The results are negative, not positive, right? It's not the experiment didn't work. It's that the experiment worked. It's that the results are negative. But very often, the negative results are just as important, if not even more important, than the positive results. Because the negative results show us specificity. They show us that something works only in a particular environment. And those are often the very most important findings. So therefore, every negative result that you can find is important to point out as well. So, for example, when looking back at the differences between witches, okay? So there was no difference in bad witches. Let's say there are no gender differences between bad and good witches, okay? I think this slide is slightly sexist, so I apologize for that. But there are no gender differences between bad and good witches. But it's very important to point that out in results to show that not everything is different. There's some things that are not different. So you want to be able to point to, very important to point out negative results as well. Only use the word significant if you're talking about statistical significance.
Mhm.
Okay? People, if you say there was a significant difference, but you didn't do a statistical test, I guarantee you the reviewer will, not be very happy with you. And for good reason. It's not only because of the reviewer. So please only use significant for statistical significance. Again, like I said, reserve information for exactly how you did what you did for the method section. That should not be in the results. And reserve comments on the meaning of your results for the discussion section. So don't go into any in-depth analysis of your results in the results. That should be in the discussion.
Ivan, can I have a question, please? When we are talking about things that are significantly different in statistical like this or discriminant test, should we always use the word significant, or we can just, for example, say that A is bigger than B or something like that?
Again, I don't think if I understood your question correct, I'll say your question back. Should you always use the word significant?
No, actually no.
What was that?
Not sure. I don't use it always. Like sometimes
Right.
Okay.
That's right. So if that was your question, then the answer is you're right. You don't always have to use the word significant. If you use the word significant, that should mean that it was statistically significant. Don't use it if it wasn't statistically significant. But if it was statistically significant, it doesn't mean you have to use the word significant. You can just say there were increases, there were decreases. it would help, and I actually do promote the idea of citing the p-value within the results section, as you said before, within parentheses. It's not every journal will mandate it, but I do think it is a good idea because it actually strengthens what you're saying. If you say we saw a 50% increase in, I don't know, vaccination, then you actually show the p-value in parentheses, it definitely strengthens your finding. And usually if you put the p-value, you usually also name the test that you used to create that p-value as well. So if it was a t-test or a two-way ANOVA or whatever statistical test that you used, you might say two-way ANOVA p equals 0.02, whatever.
Okay.
I don't think I've seen that done.
Hmm?
The results section mentioned the actual statistical test that you used.
You put it in the method.
Right.
Yeah.
But you put like a general description of methods used for
No, but in the
nominal.
in the methods, you have Evan, the data analysis and the statistical approach you would describe in the methods, right?
Absolutely. So the statistical methods must be described in full in the materials and methods, exactly how you did it. And what I'm saying in the results, so in the results, you don't say we use two-way ANOVA to show differences. That you describe in materials and methods. However, there are some, I'm trying to think, maybe we go back to some more papers. I think there are some examples I can show you from other papers where there's some where they demand that you write what statistical test you used or what your p-value was in parentheses after you mentioned what you did. So in other words, not as a full description of your methodology, but rather just shortly mentioning what your p-value was. Most journals do not demand that. And so you can write for most journals your results without mentioning what statistical method you used. Now, I'll mention another thing. Let's say you're doing a type of analysis, and it's actually a very novel or complex analysis, which not many people are aware of. So I do often write in a manuscript, and you bring as an example, saying for the next analysis, we use the blah, 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 WGCNA platform because it is something which is so novel and because it's something which is so interesting, then you can bring it up and introduce it if it's something that most readers are not familiar with. However, if we're just talking about the normal bioinformatic analysis that you probably learned about until now, an ANOVA, t-test, linear regression, whatever, then you don't have to mention it in the results. You can just describe it fully in the materials and methods. Does that make sense? Any more questions?
Mhm.
So I'll go next to the discussion and get to the materials and methods afterwards. So in introduction, you have introduced what is your research goal, or you've given some background, what's the unknown knowledge, what are you going to introduce. In the results, then you go through and you describe the different things that you found and the relationships between them. And in a discussion, you want to bring it back now from your scientific question back to the general research field. So I'm giving here an example layout. This isn't like, Torah Sinai set in stone or something like that. But this is a general example. So oftentimes the discussion will start with a very short description of main findings. And I mean very short. The main problem people do in discussion is that they oftentimes just repeat all the results again, okay? You don't need to repeat all your results. They're found in the results. So you may want to start, let's say your first paragraph, with a short description of what's the main finding that I found here. So they summarize in a few sentences, okay? Afterwards, I describe, how does this fit in with previous knowledge? Is this the same as what other people found? Is it different? If it's different, why is it different? What are the differences between my study and their study? If it's the same, great, then you can just describe the similarities. So how does my knowledge fit in with previous knowledge? The next part, and very important, very, very important, is what are the limitations of my current study? Okay, every study has its limitations. Doesn't matter what. Maybe there is a gender bias. Maybe there wasn't enough people for a very good power calculation. Maybe whatever, you know what is the limitation of your study. And you have to describe those. And we'll go into that in a minute in more detail. And then we go back to, okay, we had unknown knowledge. Now, how does this, my new knowledge, help move the research field forward? And perhaps what are the next logical research studies that can be done? What's the next logical steps? Now, some people don't want to write the next logical steps. Why? Because they want to do it in their lab and they don't want to tell everyone else. but in generally speaking, it would be nice if you can give some general ideas of what's now that we have this new knowledge. So what's the new questions that are generated by this new knowledge? Then some sort of final statement of main conclusion. So I'll go into this in a little bit more detail. As I said before, in bold and red, do not simply repeat what is in the results. Also, do not exaggerate or make extravagant claims. If you exaggerate, I guarantee you the reviewer, and for good reason, will say, why are you exaggerating? That's not true. You're exaggerating the results. And there's no reason to do that. Another important thing before going into examples is a notice there is no mention of figure numbers. No, you do not reference your figures, not reference your tables in your discussion. You did that in the results. You're done. You're done going over describing your results for the sake of describing results. There's no mention of figure numbers or statistics. One possible exception, there's always an exception, is sometimes people make a graphical figure to summarize the results. And it's not actual results, it's like a picture of as a graphical explanation of the results. You can reference that in a discussion. That's fine. You don't reference your figure numbers of results and statistics, etc. The discussion is not the place to explain the results, but to discuss the results. Actually, go through that. So I'm giving an example here of the beginning of a discussion taken from a different paper. And so here they're beginning, this is the very beginning of the discussion section, and they are very briefly summarizing their main finding. In this study, we investigated whether beta-selectin, VLM, and interleukin-6 play a role in the vascular inflammation associated with atherosclerotic disease, or just the markers that reflect vascular inflammation. Our results show that in mice, IL-6 appears to play a role in vascular inflammation, and B increases the development of atherosclerotic lesions. So in 2 sentences, they describe what is their main finding, what they found in their results. That in mice, IL-6 has roles in vascular inflammation and atherosclerosis. Fine, and there they are done describing the results. Afterwards, they go on to compare their results to previous studies. And here we have a good example. Smith et al. Previously reported that in vitro exposure of cultured smooth cells to IL-6 does not elicit the release of beta-selectin. So they state a previous paper which showed that in cells that gave this molecule, IL-6, and it did not cause secretion of another molecule, beta-selectin. Fine. In our paper, we were able to elicit production of beta-selectin when mice were injected with IL-6. Said, in our study, we were able to cause the secretion of beta-selectin from IL-6. So we get opposite findings from the previous study. Okay, they didn't get the secretion of beta-selectin, we did. So now we have to describe what's the reason for that. So they give a possible explanation. You have to give possible explanations for why your data is different than previous data. It is known that the vascular endothelium must sense both a hemodynamic pressure change and a modulation in signal from receptor protein binding before musculovascular changes occur. This combination can only occur in vivo. Therefore, the difference between our findings and theirs might be due to the in vivo nature of our experiments. So their explanation is the previous paper did this in cultured cells. They found no results or different results. We did it in mice, an in vivo system, and we do get a reaction. So therefore, the difference in our findings is because we used an in vivo system. They did not. That's why we have differences. So that's a possible explanation, and that's fine. So in a discussion, very often there might be even a few paragraphs where you compare your findings to previous paper's findings and explain why your findings are different or similar to other previous findings. That's the general idea of this part of the discussion. And then the next part might be to discuss limitations of your study. So why is this so important? And this is actually very important also if you want your paper to be published, and explain why. Let's say you had a limitation. Let's say you were studying diabetes, but all of your samples came only from came only, again, I'm using gender as an example here, but only from men. Okay, you only have male samples, and then you have some results, and you make some claim about the biology of diabetes. So you must be sure to say in your discussion, we realize one limitation of a result is that we have only male samples, therefore we cannot extrapolate to what might happen in the female population as well. If you do not, first of all, if you do not say that, the chance of your paper being accepted will go down considerably, as the reviewer will say, hey, why did you choose only males? When if you say that within the paper itself and realize you show that you are cognizant of the limitations of your own paper, then you say, okay, you realize there's a limitation, and you realize that it can't be extrapolated. Therefore, we can still allow this to be published with that limitation in mind. In other words, you have to be very forthcoming and very truthful about what are the limitations in your study. This is also important for scientific reasons and also important to be published as well. So you want to be able to be very, and the problem is very often that people, when they write the papers, they don't want to be very forthcoming about what the limitations are. They don't want, you're afraid that, people know too much of limitations. No, you should write the complete section about what are the limitations in my study and what can be the problems and the limitations and what can be done in the future to mitigate those particular limitations. So when a researcher shows that he is very forthcoming with the limitations of his research, it's a very important aspect to have. So that should be a part of the discussion. Just give one example. So this is coming from that same paper. That same paper said one limitation of our study is that experiments have been conducted only on mice. Okay. So you said, okay, here's a limitation. But then they say afterwards, however, the results for many studies of atherosclerosis, such as those examining the effects of cholesterol-lowering drug therapy, they originally performed in mice have subsequently been extended to humans. So our limitation is that this is only on mice, but we do know that in the past, some mice studies have been translated to humans. And then they go on through the whole paragraph explaining that effect. So showing limitations in discussion, very important. So that's the general idea of discussion. Now, discussions, again, every journal has its word limit on how many words can be in every section. But discussions tend to be longer than introductions, and they should be. Because in a discussion, you should be going more into detail about your findings and how they relate to earlier findings and your limitations and what should be future plans, etc. So a discussion is where you have more of an ability to be a little bit longer in your writing and to develop fully the idea of what we have learned from your results and how this helps understand, or to, as I said before, to bring it back to the general research question. Are there any questions about the discussions?
No.
No. So go to the materials and methods. So the materials and methods, as we've said before, is we need to provide all of the information necessary in order for an independent laboratory to completely perform your work. Therefore, you need all the information, including exact amounts, concentrations, even where the reagents were bought from. In materials and methods, you should include as much, all of the information. In other words, I should be able to read your materials and methods, and if I have the type of lab that does that work, I should be able to do that exact thing in my lab. That is the aim of materials and methods, to produce reproducibility. Someone else can reproduce now your results by doing the exact same thing. Someone else has to know exactly what you did. And that means including even where you bought your reagents from. Where does everything come from? Unlike other sections of an article, materials and methods should be long because you need to have all of the information there, unless it's a type of work that doesn't need much materials and methods. But the general idea is don't be afraid of it being long. On the contrary, because of the necessity, it's right here, to have materials and methods being very long, but you don't want to publish so much in a small journal, what many journals now do is you have your materials and methods published as supplementary material. So very often you'll have your journal paper, but then there'll be a link to the supplementary materials and methods section, where I'll have even a longer explanation of all of your materials and methods. So you don't have to worry about making materials and methods. What was that?
So they have like an abridged version of the materials and methods in the actual article, and then a link to the full version?
Right. So that very much depends by journals. In most cases, what they'll do, well, if your materials and methods can be fit into the journal paper, then they'll just put it there. But if it's too long, then sometimes it's completely dependent on journals. Sometimes they just put the whole materials and methods. But there are also other times it's right where they have it abridged. We can actually, if you remember, there was that PNAS paper, which I showed you before with the oxytocin. There they had an abridged version on in the paper, and then a link to the full version. So that's true. Sometimes there'll be an abridged version in the paper and then a longer version online, but it's very dependent on the journal and their requirements. A few tips about materials and methods. They are always use the past tense, and they are also written in the passive tense. As we talked. Again, I'm hearing voices again. Maybe it's my problem. I need the psychiatrist again. But
Evan, can you hear us?
Yeah, I can hear you fine.
Oh, we just lost the connection. Just to come back to the length and the appendices and stuff. Most people here are doing clinical research, so it's I think in lab research it's maybe more common when you have, for example, a very detailed lab protocol. Right. You may not want, but a good example is with questionnaires. Like in the materials and methods, you don't want to, if you have 250 fields in your questionnaire, I've seen it before. People say, in the methods, we recorded information about blah, 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 and there's a list of like 250 fields. You don't want that. You just want, the big categories, and then you can say, see Appendix 1 to see the whole questionnaire, for example, or something like that.
Okay, that definitely makes sense. That definitely makes sense.
I would think that if I read an article, I mean, probably we're talking about more lab research and stuff, but when I read an article, it's going to be kind of weird not having any explanation as to the methods and then having to look for somewhere else.
No, you have to have the right balance. You also don't want to like bore your, like for example, if you describe a lab protocol which says, you pipette 2 milliliters of this into that, and you don't need pages unless if you're really, really interested, you can go and read it. But most people are not going to be interested in that level of detail. But it's important for it to be available.
That's my point. You have to have something.
Oh yeah, absolutely.
So this might be the sort of thing which is different between, as you said, clinical and or hardcore lab biological research, because for more biological research, it's actually very important to include how many not necessarily microliters, but volumes, concentrations, everything, and that's why it usually is as an attachment. Because all those things are very important to understand, to have someone else reproduce your data. So very often people actually don't really read the materials and methods unless they're looking to understand completely the protocol. And this is the fact, is like if someone just wants to understand the biological knowledge, most chances they aren't even going to look over the materials and methods. Maybe in clinical research, it could be slightly different. So I realize that. So I give this example. This is, for example, again, this might be more relevant for biological research. But if this, actually I'll go to a forward example. Let's say this is a protocol that you used for making a solution. In this case, a solution of EDTA, right? So you dissolve EDTA and water, and then you stir, and then you adjust water, et cetera, et cetera. So the way this would be written in the materials and methods paper is the following. You do not write it as a protocol, but rather what was done in a passive past tense. So to prepare an EDTA stock solution, the following protocol was used. First, 186 EDTA and blah, blah, sodium hydroxide dissolved in 100 milliliters of water. In other words, all of this, so at least in biological studies, all of this is definitely included in the materials and methods. There's every detail. Because the reason is, for example, there have been many protocols where I have taken materials and methods from other papers and just repeated that same thing in my lab. And you have to have the ability to do that. And if not, what will happen is other researchers will start contacting you, asking you exactly how to do this, how to do that. And unless you want to be bothered all the time, then it's best to just explain it all in materials and methods. It might actually even be interesting. Just 1 second. Maybe I'll go back for a second. Do stop share screen. Go back to the papers we're looking at before. When we look at the so this paper, this cell paper, is very much a biological paper, right? In mice. So they have a lot of data in mice. So if I go to the end of this paper discussion, experimental procedures. Okay, look at this. Experimental procedures. See supplementary information for additional details and references. And then they have here this short explanation of a few of their protocols. See, that's actually, I don't even know why they particularly chose those protocols, but they chose a few, then describe them here. And again, when you're reading these materials and methods, you can see fecal samples are collected at 2 weeks after gavaging mice with blah, blah. Bacterial genomic is extracted from the fecal pellets using the stool kits. You have to say what kit you used, where did you buy it from, et cetera, et cetera. But however, this is actually a very short explanation of their methodology. If I were to click on this link, this supplementary information, I think what I get is the following. Just 1 second, I'll find this here, right. cell paper with materials and methods. So here I get the same paper, right? It's the exact same paper we're looking at before. I'm going to go to, and now here, so I get the same point, experimental procedures. I know you might not see this as well as before. Experimental procedures. And now afterwards, and now I have references. Now afterwards, extended experimental procedures. Let's see how many pages I have of this. S1, S2, S3, S4. So the extended procedures were 4 pages. So they have 4 pages here describing all of their other procedures and exactly how they did all of them. Again, you have your exact concentrations of antibodies, concentrations of solutions, et cetera, et cetera. If we look at the paper with the treatment of people with oxytocin, so this is people with, and it'll go again to materials and methods at the end, discussion, materials and methods. So this here, it looks like, so I'm going to look at here. Okay, so this is materials and methods, but see, this is only part of it because they have here what they call here SI, supplementary materials and methods. But in what you put in the paper, they have about 2 pages of methods. And I'll even try to go a little further into this. A group of 13 adults with a clinical diagnosis of Asperger's syndrome or high functioning autism, according to were recruited from expert centers. Actually sent to Professor Edelstein this paper as well, so maybe you can see this as well. But this is an example. Again, it looks to me like they are also going very in depth into different parameters of these individuals that they took into the study. Behavioral experiments, exactly how they did the behavioral experiments. But you also have here a supplementary materials and methods. If I click on this. Supporting information, 1, 2, 3. So that's almost 3 pages of supplementary procedures about how they did what they did. They even show here algorithms for the different computer programs that they used. So they also went really in depth, but again, for the really in-depth stuff, they put it on supplementary materials and methods. So I'll go back. Sure.
חייבים קפה, אני לא...
Evan, can we take a 10-minute break?
Sure. The truth is also, I don't have that much longer as well, but we can definitely take a break.
Okay.
Mhm.
Okay, so we'll take a, I think, I can see people's attention needing coffee. We'll take a few
No
minutes.
problem. No problem.
Okay, great. Thanks. Evan.
So let's say until 11:10.
What's the time now? 5 past maybe. It's
5
10:57.
past.
That's 8
No
minutes.
problem. 5 past. And I already see all of the coffee and the food, and I'm
We have
jealous.
salad. Yeah, we have the administrator of the department is also a very good cook, so she made salad and everything, which is very nice. This is what she did. If you were here, you could.
I know. I'm jealous. Cool. And let me see if I'm shared screen right now. Stop. Share. Okay. So we're talking about materials and methods. And just two other short ideas about materials and methods is that sometimes, let's say I'm using a protocol or a methodology or even a questionnaire that someone else used, that someone else developed. So I will also describe that in materials and methods, but beforehand, I will mention or reference where this protocol was previously described. So just give an example at the bottom of this slide, where we did a particular protocol that we had done in a previous paper, and we described it in a previous paper. So I said, we performed this experiment as previously described. Then we give the reference, but I still have to describe it in this paper, but I could do a slightly abridged version. If there is, let's say, like a very long description, then I can very much shorten my description here because it was published somewhere else. Let's say this wasn't my protocol, but I'm now using a protocol that someone else developed. For example, we're using a protocol in a lab, doesn't matter, called an intact protocol. So I would say in the beginning of that section, materials and methods, we are using this intact protocol as previously developed by Boris et al. And then I'll give the reference. And then I could also shorten my description because it's been described somewhere else very well. Unless I made any changes to that protocol, then I might say, I use that protocol as described there with the following, just describe my changes. So in other words, if there's something that's a very long protocol, but it's already been published, I could just give a short and abridged version of that protocol in our paper, but reference the previous one where they can find the full protocol. One thing which someone else did mention before, statistics, there should be a paragraph about statistics, just mentioning what were the statistical tools you used throughout the manuscript and when, if it's ANOVA, t-tests, what programs you used, SPSS, GraphPad, et cetera, everything, in words, all the information that's possible to give, then you should give. One other thing that I just wanted to mention you should know about, again, this might be slightly relevant right now, but will become more and more relevant as the years go on, is that some of the major journals right now, before you publish, you have to fill out a checklist showing that you have shown all of your materials and methods. So I'll give one example. I have a few examples. I'll give one that I can show here of such a checklist you have to fill out when publishing in a journal. And 1 second, I'm going to share screen here, share screen, share. And go to, I think it's down, reporting checklist. So this is for a journal called Nature Neuroscience, which is a very major neuroscience journal. It's very top notch. And you have to fill out this checklist when you submit to them, which includes, everyone can see my screen, right?
Yeah.
Which includes here, for example, in every figure, what statistics you use, which test, what was your N, what was the number of samples you had, descriptive statistics, p-values, degrees of freedom, all sorts of statistical and fun stuff. So here I was actually one filled out, the NOVA test, what were the p-values, et cetera, long explanation. Is there a justification of the sample size? If so, how is it justified? Are statistical tests justified as appropriate for every figure that you have to fill out here on the right side? So here's all sorts of questions about statistics. We won't go into all of them. One other thing, which again, you should just note, have you, I don't know, talked at all about figures and about different types of plotting of figures? Maybe not.
Yeah, we had a lecture on figures and
Figures.
and tables, yeah.
So there's some journals, I know all cell journals, cell reports, nature journals, etc., that they have stopped allowing bar graphs to report statistics in the papers it publishes. What that means is that when you report the graphs, you know, there's oftentimes bar graphs. You have like a bar for your control samples and a bar for your treated samples or whatever. Then they, instead of showing just bars, you have to actually show a dot for each individual within the group. So that's one thing they point out in this paper, in this checklist. And so here, you not only have a checklist for statistics, we have a checklist for reagents. If antibody has been validated, what are the cell lines that have been used? Where is the data available? This is another thing which I don't know how well, how applicable this is to everyone to the room, but it's something that you should be knowledgeable about. Let's say you make data, for example, genomic data, or other types of high throughput data. Very often the journals demand that you make the data available to the scientific community. Again, I think this is somewhat, might be less applicable to everyone in the room, but if
it is applicable because journals, even clinical journals, starting to demand the data sets and, you know, anonymized, basically to promote reproducibility, transparency. So it's very, and the checklists also are becoming very, very common. So you'll see they're a bit different, but, you know, things like the consorts checklist or the equator protocols, which go through each of the different parts of your manuscript, and you have to demonstrate that you've done all of these things. Yeah.
Excellent.
It's very similar.
Okay, so it's a similar type of idea. So for example, for the data availability, let's say if you produce some sort of data, it could be clinical data. There are often online repositories where they demand that you put your data in one of those repositories. Just an example, when we make genomic data or RNA-seq data, we have to put them onto a online repository called Gene Expression Omnibus. And then we have to, in the materials and methods, we have to give a sentence of exactly where our data was reposited. And not only that, but actually I even tell a story in we have a paper which is currently in revision. And when we wrote the paper, so actually I made the mistake, I completely forgot not to put, I didn't put our RNA-seq data, our genomic data into one, actually no, we did put it in a repository, but I forgot to give some of the basic statistics in the materials and methods. I didn't think about it. I'd read to you the reviewer I got back from the paper I sent, which is all about transparency of results. I want you to show me this raw data. All of your data should be put into this repository or that repository. Right now, people are very, very stringent. Also journals, also reviewers, on complete transparency of data so that other people can, first of all, reproduce your data. There's also another reason, which is particularly true, at least in genomic research, other types of research, is there are many other researchers who will take let's say, data from 10 different labs that have already been published, then take all that data together and do a reanalysis, let's say a meta-analysis or something like that. So another reason why all raw data should be published and made available. And so usually it's demanded that the data be made available by publication, and many journals demand that they be available upon review, so that the reviewer can go inside your raw data as well and see what it looks like, etc. So that's a very important point. So you have, again, this sort of checklist. Second, go back. If we go down here, even here, they'll talk about what computer codes or software was used. And here is about the human subjects, about your IRB, demographic information, number of human subjects. So this has more, maybe more clinical applicability. About questions about conformed effect and consent, groups being matched, patient photos, and then you have further things for fMRI, functional MRI studies, et cetera, et cetera. So this is a general idea. This is something that is becoming more common, that you need to fill out these checklists to make sure you have all of your data both available, and also that the statistical analysis was done in the most appropriate ways possible. This is another important thing to keep in mind. I'll go back. Okay. So that was about the materials and methods section. Now, the only two things I have left to talk about is a short explanation of abstracts and of and a little bit about submission of papers. Just so I know, has there been any previous discussion of abstracts in this course?
No.
The answer was no?
The answer is no.
No, okay. So abstracts, first of all, I think they're actually the easiest thing to usually write. It takes a few minutes. And the reason is it's basically a very short, concise version of all of your abstract, of all of these sections of your manuscript. There are usually two types of abstracts. And a journal will ask for one of these two types. One being a structured abstract with subheadings, or what you just call free form. So I'll give an example of a free form abstract. So every type of journal, we usually ask for one of these two, and you have to see which type of abstract the journal requires. So this would be a free form abstract. I actually took from a paper that we published. And when we, of course, when we gave the abstract, it didn't have these colors here. I color coded them now so that you can see basically the different sections that are in this abstract. So in here, the first three sentences are basically introduction. Here you see CTCF is an organizer of higher chromatin structure and the regulator of gene expression. Genetic studies have implicated mutations in CTCF and intellectual disabilities. It's introduction in red. And then one sentence of what is unknown or what I want to show or what is the hypothesis. However, there is no knowledge of the role of CTCF mediated chromatin structure in learning and memory. That is my sentence of either unknown knowledge or hypothesis. In other words, what are we asking in this manuscript? Everything in blue here is basically a short and concise description of the results. And then what I see in sort of a dark red is the conclusions. So that's an abstract to basically have an introduction, results, and conclusions aspects to it, even if it doesn't have subheadings per se. Notice that in an abstract, there are no references. Because if you said something in your introduction, if you said something in your abstract which needs a reference, you will surely repeat it in your introduction and there give it the reference, the appropriate reference. So there are no references. You don't talk about what's in figure 1, 2, 3, 4, or 5. It's just a brief abstract explanation of what will be in your paper. So that is the general idea. And that's in the free form version. There are some journals which will ask for a structured abstract. So I'll just show you an example. This is an example I'm showing you now the introduction, then the methods, then afterwards you have the findings and the interpretation. Now again, every journal might ask for different subheadings. Sometimes it's just the same thing, but they call it different names. Interpretations could be conclusions or whatever. Findings is basically results. But those are usually the major sections that are found in a structured abstract: introduction, methods, results, conclusions. You will see that usually structured abstracts are a little bit longer than the free form. So if you remember in the papers that I showed you this morning, when we saw for a minute the abstracts, they were all free form. And those are because those are journals which ask for free form and they want short abstracts in a free form without any headings. Usually structured abstracts will be longer, but again, the journal will tell you what is your word limit for how many words you can put in. And if so, then again, you can start with your 3-4 sentence introduction into your field, which is basically a shortened version of your introduction, then methodology. And again, methods is not going in depth into the methodology, but is just giving and giving any big details necessary to reproduce the results that you'll find in materials and methods. But again, is to understand the main methods being used. So for example, if I read this, we developed an egg-independent strategy to combat the avian influenza virus, because the virus is highly lethal to chickens and the maintenance of a constant supply of aboriginated eggs would be difficult in the pandemic. We use replication and competence human and viral vector-based hemagglutinin subtype 5 influenza vaccine. I have no clue what that means, but I'm sure someone in vaccination understands what that means, which induces both humoral and cell-mediated immune responses against avian influenza viruses isolated from people. So again, that is an overview of the methodology used in the paper, followed by what were the main findings. Again, 3 sentences, 4 sentences of the main findings, very similar to what you might find in the very beginning of the discussion that we talked about before. When we talked, so at the beginning of the discussion, you want to have a few sentences summarizing the findings. Same thing here. And then followed usually with some sort of conclusions, interpretations, where again, you take whatever results you have and you bring it back to the original research question. Say, okay, so in this case, our findings highlight the potential of an AD vector-based delivery system, which is both egg-independent and adjuncent-independent, and offers stockpiling options to development of a pandemic influenza vaccine. So this is why our results are important for the general research field. So again, the interpretation is just to bring it back to why you did this experiment in the first place. But again, this isn't a place for discussion. You know, we don't talk here, and don't talk here about limitations and previous papers and stuff like that. But basically, just what is the meaning of our results to the research field. I just mentioned a few things here, even though this might not be the particular goal of the session, but abstracts are very often very different for conferences than for manuscripts. And this might be relevant if you've had to write before an abstract for conferences. You might have it might have been important to write a longer abstract so that they can understand your work and decide if to allow you to speak at the meeting or to give a poster at the meeting, etc. So they usually want a lot more information in an abstract for a conference. Well, in an abstract for a manuscript has a very different goal, which is basically an overview of the paper, because all the important information will come in the manuscript itself. Something to keep in mind, because sometimes people who are used to write it's common for them to write an abstract for conferences. So they'll write one for a paper, but just is too long and too much information, because that's what you write for conferences, but not necessarily for a manuscript. So it's just an important thing to point out. Abstracts for manuscripts often have a limit of words between 150 to 500. That's the usual limit, somewhere in there, which is why we have to keep it short and to the point. And just get your main point across. Again, what is the main point of your story? What is the thing that you want someone to get away from your story? So that should be the main focus of your results and the conclusion of the abstract. So with that in mind, I thought I would just look at an example maybe of one or two journals of what they ask for when submitting a journal for publication.
Evan, can I just say one quick thing about abstracts?
Of course.
And I'm saying this as a journal editor because I see a lot of abstracts, and when you submit, it's the first thing that editors look at. And often what happens is you write your manuscript and you spend a lot of time and effort, and then in the submission process, you realize, oh, I forgot to write the abstract, and you write it in 2 minutes. And it's worth actually spending a bit of time, because I would say if the abstract is not catchy to the editor, he's not going to bother reading the paper. So it's really your chance to show the editor that this is an important piece of work. So don't neglect it and leave it as a afterthought. Spend a little bit of time and choosing the results that you want to highlight that really show that your paper is important to the field.
I think it's a very excellent point, and I think that was a good contrast to what I said in the beginning, that maybe the abstract is easy and it could be written shortly. So
it is easy to. It's not hard, because you don't generally you copy and paste and adapt. You don't need to think about writing new material, but it's short, and often people struggle with the length. They find it very hard. I get a lot of requests saying, "Can you help me shorten my abstract?" Because really distilling things to the core essence, like you did in the beginning with the sentence, is not always easy, particularly if English is not your first language.
I think that's an excellent point, and I think what abstracts are where that ability to use short sentences and shorten your ideas to very concise terms is where that really comes into play, and particularly in the abstract. How can I be as convincing as possible in a relatively short framework? And I'll just strengthen what you said before, which in much of your results paper, you're trying to get across points. Abstract is really where you're trying to sell the idea in a particular sense, even though it's just not, you're selling an idea based in facts, but you're still selling an idea that you want people to notice. So I think you strengthen your idea that almost the abstract is sort of like the selling point of the paper. Will someone read your paper or not is basically based on your abstract. So I think it's definitely right.
Great.
If we go on to shape. There's one other thing, by the way. Actually, I'll just mention that I didn't put any slides, but it's along actually the same point or of what Michael brought up right now. Is there's actually something else that you do also when you submit the paper, and it's what you call your write a cover letter.
Yeah.
You actually write a letter to the editor of the journal, and in that letter as well, you also describe what is what's in your paper, but again, this is the selling point, trying to say, why is what I have to offer so interesting to the scientific field? And since maybe Michael, scientific editor, maybe you can speak of this even original editor can speak to this even more. But all I can say from that is that usually this is a letter. When I write it, usually around half a page or so. I just try to get the main. I say there's a really interesting idea that is unknown in science, which is X. And now what I am showing you is how something really interesting new data that can really help solve that question, and what we have found is why. And so it's a way of just trying to convince the editor that what we found is really of interest to the scientific field, because there are some journals, I already start explaining this now, dependent on the journal, there's some journals that will only send a very small portion of the papers they receive to external scientific review. In other words, you have now submitted your manuscript to journal, I don't know, journal of exciting stuff. And this journal will only accept at the end maybe 10% of the manuscript submitted to them. Therefore, they don't want to send every manuscript they receive to external review. Therefore, they'll only send those papers that they think are of real interest and think are within their journal's interests and within the general scientific interest. So both the abstract and the cover letter are actually really good opportunities to try to convince the editors of the journal that what you have to offer is actually really interesting, is actually really important to the scientific field. So that's something that is actually a really important thing. I don't know, Michael, if you have something more to add on that.
Just say my rule of thumb for letters is that it shouldn't be more than a page. If you're not able to explain in a page what your paper is about, then it's probably not that important, or you need to improve your writing skills. Because again, you have to, like, you know, as an editor, we probably send, I would say, 30% of the papers we receive to reviews, and then the reviews end up rejecting 60% or something. So 2/3 of the papers we get,
You don't even look.
we look at, but we don't go to the, we don't send them to reviews. We reject them at this stage. So there's two reasons for that.
Mhm.
They have not enough scientific merit, or they're really badly written, and often they're really badly written. And you just don't have the, you know.
Are you always candid about what type of, which of the two it is?
So generally, if you reject, you don't always give if it goes to the review stage, you'll get feedback from the reviewers. If it doesn't make it to the reviewers, it depends. Sometimes we do, sometimes we don't. It depends on the time, you know. So,
at one time I got rejected. This article got rejected. It's nicely structured. It was written well, but it's not. I don't know. It's not within the exact scope of a journal, something like that. So is that like a standard?
No, it depends. Sometimes it depends. Sometimes if we feel that a paper is important but not well written or not the right journal, we'll generally write and say, you know, you probably need a more, like, you know, it's an epidemiology journal that I'm a deputy editor. So we say, you know, you should send it to a clinical journal, or, you know, if it's or to a national, you know, like a national journal rather than a global journal. If there's nothing wrong with the, you know, sometimes you have very good papers that is well written with good results, etc., but they're just not that relevant to a global audience. Maybe they're relevant to a national audience. So you tell them, you know, go to a national journal. If it's really poorly written, and you can also see how much effort sometimes it's a language issue. Sometimes you can see that the authors just didn't put that much effort into it. And if, you know, if they don't put a lot of effort into it, I don't put a lot of effort into it either. You know, if there's typos, if there's all kinds, you know, there's really, really interesting stuff, you know, sometimes you can see really bad stuff. It happens a lot. So it depends. Sorry, Evan.
No, not sorry at all. I definitely I would agree that as a reviewer, I sometimes have to review quite a few articles in terms of English and writing skills. So very often rejected quite a few papers. I think the the number one reason which I've rejected just outright manuscripts is because the writing or the presentation was just like way out there or wrong for some reason. That's just like the number one reason. In terms of when you say, someone said there about the honesty or about, it's sort of like that the real reason why they rejected your paper. So I've had a few papers rejected where they didn't send it to external review, but actually they gave a really good, I don't know, not very long, but then let's say 7-8 sentences and sort of some internal review, something the editor said, we thought that this part of the paper was strong. We thought that this part of the paper was weak. I've gotten some things back like that, and when the editors put in that sort of effort to show strengths and weaknesses, I definitely think that that's something which is quote unquote believable. Yeah. I'm going to just 1 second share screen. Cheers. So if I go actually to my internet, right. So just give an example, maybe you've seen something like this before, but when I go to Journal of Neuroscience, and I go here, I want to go into this paper, into this website, and I can go to for authors, information for authors. Basically, any journal should have this readily available on the website. And here I have basically complete information on, usually more information than I need on what exactly is the outline of this particular journal for papers. So here they have lots of policies on copyrights, and I'll go through that, but preparing a manuscript. So here they tell me exactly what should be in every page, what should be in the title page, the title, abbreviated title, authors' names and affiliations, et cetera, number. They want me to know the number of pages, number of words for an abstract, introduction, discussion written on the first page of the paper, acknowledgements, et cetera.
Yeah.
Did someone say something? I can't quite hear.
People they're just complaining about the requirements.
Yes.
But it is true. The best, the truth is, when you see these, sometimes you really think, they make your life difficult. Like, for example, the Lancet journals, for some reason, they want the dot in the middle and not the dot in the bottom. Like, you would think that the industry could standardize these kind of things. You get paranoid, and also it gets worse. It gets worse and it happens more than you think. For the same journal, the different parts of the instructions are contradicting each other. But the best piece of advice is read these and comply, because they will not bother if you didn't bother going through the requirements. They will not bother reading.
So sometimes when you prepare a paper to be published according to the instructions in one journal, and then you want to, and then it gets rejected, you want to try another place, and you got to reroute the instructions and then redo
from the start.
It's like, you don't like start from the beginning. You have to re-edit it again. And
no, it's a pain, but
yeah. So you have to be obsessed.
Even if you weren't before, you get obsessed.
Yeah, it is.
אם יש לך עוזר מחקר שיעשה לך את זה.
אתה חייב, אתה לא כמה שעות תעשה.
It takes hours. From having the manuscript to like the editing and the preparing for submission takes a few hours for sure.
For you it takes a few hours. For me it's going to take a week.
That's a lot for who is experienced. It's a lot, but you have to do it. So you can choose also journals that are less stringent, and then they insist on British spelling versus American spelling.
But if you have staff and someone that's what he does well, writes well for any script.
Well, some people do this for a living. You can actually pay someone to do it if you have the budget. if there's, you know, people who are like writers and medical writers and stuff.
Huh.
But it's not cheap.
No, but who? And how much does it cost?
It's not cheap.
So yeah, I just note again here in Journal of Neuroscience. So for example, they give their, how do you say, the definition, they want 250 words maximum, which is, I think many journals somewhere in that area for an abstract. There's some like nature journals, which I think are 150 words maximum. They're very short. So there's the general ideas in the introduction, 650 words, including citations. I'm actually interested in what they give a discussion here. Experimental design, blah, blah, code software. Discussion, 1,500. So you see that difference. Introduction is 650, discussion is 1,500. And it's usually in that ballpark. The discussion will be at least twice as long as an introduction. So that's a general idea. And again, they give their how to do references, but references everyone does today by different reference making tools, which you're probably familiar with already, and etc. So again, as Michael said before, the best thing is to go directly into the journal. And and this is why I always, before you start writing, I always decide what journal I'm going to submit to before I start writing. So at least the first time we don't have to change anything. But if we submit to somewhere else, then you might have to change something afterwards. That's right. It's usually just the abstract length or something like that. Let me see. That's pretty much the outline that I prepared to talk about today. I don't know if there are any questions, issues, etc.
Maybe suggestions on the interplay with the reviewers. But how does it work? You get a letter with the
reviewers.
remarks, and then how do you approach it? I mean, do you have to?
So I'll give my input on that. So in terms of let's talk about so first you submit to the journal. And depending on what journal, depending on the level of that journal, you'll probably get a mail back. Could be within 1 week, could be within 3 weeks. It depends on the journal and their backlog, saying whether or not they've sent to external review. And why or not. If you sent to external review, they'll just send you an email saying we're happy that you sent to external review. If not, then they may give reasoning why they did not. Usually at that stage, there isn't much to argue with them if they haven't sent to external review. So after that time period, they'll send to external review. Again, complete depending on the journal, you'll probably have to wait like a month, 2 months, whatever, until you get your reviews back. Usually being either 2 or 3 reviews. Today, I think, I don't remember the last time we received only 2 reviews, and they're usually 3 reviews. Most papers have sent in today. And at that point, again, I think you already know the technicalities when give some advice. So in terms of technicalities, you usually get something back being either minor corrections, major corrections, or not accepted. I'll give some of my recent experience with a journal called SILD Reports. And a few years ago, I sent to them a manuscript, went to external review, got my reviews back, and I got a reply which said, which is called rejection with the possibility of resubmission. which what we had to do is I had to write them a letter explaining how I would reply to each of the reviewers' comments. And then the editor would decide if you would allow me to do those experiments and give a revision or not, or he'd just stop it there. At which point I wrote like a 3-4 page description of exactly all of the reviews that all of the changes we would do to the paper, all of the new experiments we would perform, et cetera, and allowed him to do it. First of all, I think this is a common knowledge, but first of all, always make sure to respond completely to all of their comments, all the reviewer comments, and of course, the most cordial manner, responsible, even if they said something completely ridiculous, but to respond as cordial as possible, but more so as thorough as possible. There's sometimes we've had to explain to reviewers that an experiment is either impossible or was not possible. And that's acceptable, that's fine. Most reviewers and editors can understand that if explained in a well thought out and meticulous manner. Sometimes reviewers will request experiments, you just have to say that is not possible or was not possible for reason X, Y, Z. And you have to be able to give a good explanation of that. And so I've done that on quite a few occasions. Usually when I get back again, this might be slightly different because of biological papers versus clinical papers, but I'll usually get back reviews with, let's say, a list of, let's say, 3-4 experiments they would have wanted me to do. And I'll explain to them why we did 2 or 3 of them, but there was one that couldn't be done for whatever reason, and that's fine. Again, Michael, is there something you want to address to that?
Just to add, so the first thing is, I think it's important to accept reviews as an attempt to improve you, not to annoy you. That's the first, and I think you have different categories of reviewers. You have, I would say, 3 categories. The first one is reviewers who didn't bother to read your paper or didn't actually understand your paper. Reviewers who genuinely are making useful suggestions, and reviewers who, like Evan was saying, are basically asking you to do a different study. So I think that for me, that the way I respond to them, first of all, yeah, always be polite and always say, thank you for this great review. And, we believe that our manuscript is now much better. You always say that. Where you can make changes, you do them. If you do feel that what one of the reviews is asking is not reasonable, or you can choose not to do it. You can choose to actually say, we didn't take your suggestion, but if you do that, you need to justify why. So for example, I had an example recently where we did the review said, you should use this statistical test, you should have used this statistical test. So I went back and said, actually, we're sticking with what we did because X, Y, and Z, and we went into the distribution of the data, etc. And then he responded and said, actually, now that you explained it more clearly, I agree. So it's not like you have to implement everything. If it's simple, sometimes it's not worth arguing. There's a bit of ego as well. Sometimes people say, you should cite my paper, or if it doesn't cost you much, just do it. But if you feel that it's actually not improving your paper, then it's okay to do so, providing you justify it. And if they say, oh, if you're doing a project on ileostomies and said, oh, why didn't you look at patients with, a completely different operation? You can just say, well, it sounds like a really good idea, but it's outside the scope of our study or something like that, because sometimes people come with, ridiculous, or why didn't you collect this data instead of that? Well, the data's collected now, so there's not much you can do about it. It's okay, but you just have to explain everything, and don't ignore any points. Answer them, even if you didn't make the changes, answer them.
Is there a format?
Just an ask questions.
No, generally it's point by point. So you put the specific comment and then the reply underneath, and then you have to prepare a document that you add to the resubmission, and that gets sent to the reviewers. Often, when as an editor, when I receive a revision, the first thing I look at is the response to review. Great. We're having a fire alarm, Evan, so that might be a good natural point to.
Evan, thank you very much.
Thank you.
Thank you very much.
Thank you.
It was, really fantastic.
Hope that helped.
And I'm sure all the manuscripts will greatly benefit.
Have a great day.
Thank you. Bye-bye.
Bye.
Bye.
כן, לא, אני עכשיו עובד על משהו.